Welcome to my presentation. Today I discuss about DNA cloning with plasmid vectors. At the beginning of my presentation, I tried to give overall configurational structure of DNA cloning technology and the performance area, how we get our desired cloning DNA. Mainly, DNA cloning is related to biological technique, and the main purposes of this technique is to insert our desired DNA gene, in particular uh, vector. Then the vector is inserted in the bacterium cell. Then the bacterium culture, and then finally we get our desired product. In my overall presentation, I try to explain this gradually. Now, my next slide, I try to present the engineered and the applied area and the applied process, how we replicated our plasmid and the mainly parts that are involved in our plasmid cloning vector. In this slide, here I try to present the three parts. One is the replication of gene, another is the ampicillin resistant gene containing this vector, and this is the part where we insert our targeted DNA gene. Now it is the plasmid and the replication origin combined with the plasmid structure, and they form daughter plasmid. Now this plasmid can easily replicate it, and we get several types. We get our desired product. Now DNA cloning with plasmid vectors, mainly plasmid vectors that are selected, and by the using of restriction enzyme we cut a specific site. Then this site is or cover or site is attached with our targeted DNA. Then we applied it in and the E. coli bacterium cell. Then the bacterium multiply and we get several product. But here the main fact that the ampicillin resistant gene we can apply here, and in the ampicillin containing media, only the only the bacteria survive that contain this type of plasmid. In this figure, we can see that this bacterium is the absent of plasmid. So, in the ampicillin containing media, this bacterium is not able to grow up. By using this theory, we can easily identify our desired product, and this is very crucial thing for us. This theme here I try to present through writing the ampicillin resistant genes encode beta lactamase inactivate the antibiotic ampicillin. So the plasmid vector that containing this resistant gene that can easily survive in the ampicillin containing media, otherwise not. Now the restriction enzyme. The restriction enzyme, the such type of enzyme that are used to cut the DNA. Here the sticky int we cut the DNA. Sticky int means the equal portion. Through the equal portion we cannot be able to cut it. So the sticky int form, and then uh, our main purposes can be uh, happen when we use this restriction enzyme, but the presence of methyl group with base. That's called methylated DNA. When methylated DNA present, the time the restriction enzyme not able to cut the DNA structure because thus methyl group not allow this enzyme to do their roles. So they are unable to perform their function. Now, when we cut the DNA, but at a time we need to combine this. For this combining purposes, here the ligase, DNA ligase plays vital role. In my figure, I try to present DNA 1, DNA 2. Here several structure combined A, B, C. Now we can phosphate, hydroxyl group, and here phosphate, here hydroxyl group. They combine to each other and they form a pure structure. Now the DNA ligase help them to combine perfectly and form the better structure that are our dessert product. Through this process, finally we get such type of nucleotides, several chain reaction like monomer 1, monomer 2. They are coupling with acid and form dinucleotides. In this position, they are combined to each other. Here the 3 position and here the 5 position. Page 1 5 position combined to 3 position of monomer 2. Following this process, if we repeat this process again at that time monomer 3, 4 they are combined and at finally we get a long oligonucleotide chains that our dessert product we get finally. So it's the main purposes that are filled up through this process. Now, our overall presentation, our main purpose is to get such type of product that contain our desirable genetic code or genetic structure, and in several purposes for scientific research or developing several animals dolly, such type of sheep that are cloning in 1996, and by the using of this cloning technology and several application of this technology are for scientific research, scientific several purposes: gene expression, production of recombinant protein, production of insulin, and several hormone that are gene therapy purposes. So it's overall knowledge of this technology is essential for a pharmacist because if he or she want to do better for research purposes and if they interest to work about this gene therapeutical side, they have must do this technology, and by using the technology they can perform several types of product and they can develop several types of product or animals characteristics that are essential for future development for the scientific field. So as a pharmacist we have much knowledge about this. Thank you all. This is all about my presentation.